Boulder now.
Sorry.
That's okay. No problem. Right, so I'd like to talk to you today a little bit about the work that my lab does trying to identify the cellular basis of positive social interactions. So as humans, we find comfort in close social interactions, and when we're faced with a fearful or a traumatic situation, oftentimes our first response is to reach out to those that we love. And we know that these social interactions, such as those you can see here, are not ephemeral. They have a direct impact on our biology. Specifically, interacting with people that we love can help to buffer against the extremes of heart rate and blood pressure. And it also helps to return our stress hormone levels back to basal levels faster after we encounter a stressful event. In sort of a larger scale picture, we know that having healthy social networks can promote recovery from cancer, heart attack, and other maladies, and it's one of the best predictors of longevity in humans and in monkeys. And so the question that my laboratory is interested in is what are the neural circuits and the specific cell populations within the brain that are responsible for mediating some of the effects of positive social interaction on stress, fear, and behavior. So fortunately, this effect is something that's conserved across many species. So we can see that this buffering of stress, if we look at goats, if we look at ostriches, monkeys, humans, and rodents. And in my laboratory, what we've done is we've developed a behavioral test that allows us to directly assess how much a companion animal can reduce fear levels. And to do this, we take advantage of the fact that when you put a mouse into a large, anxiety-provoking setting where it can't run away, what it does is freeze. And that freezing is an indication of its fear level. If we take a mouse and we put it in the exact same setting, but with its well-known cage mate, what we see is a reduction in freezing levels, indicating that the animals are less afraid. We can do the exact same thing, but in an environment where we teach the animals that they should be afraid by applying a mild foot shock. And again, we see that the presence of a companion animal reduces freezing levels, indicating that they are less afraid. And so what these behavioral tests have taught us is that the presence of an affiliative companion in an aversive environment, whether it be innately fear-producing or one in which you have to learn to be afraid, reduces levels of fear. So the question that I'm interested in is what neurons are actually being activated in your brain when you're interacting with another individual, and if we could identify those neurons, come back to them later and turn them back on again, would that be sufficient to mimic these behavioral effects and reduce fear? And so we did this experiment in mice where we have some fancy transgenic tools that allow us to do this. And so in green, what you can see here are neurons that were initially activated when an animal was introduced to its new cage mate. Now these neurons are permanently activated in a way that allows us to employ optogenetic techniques, shine light on them, and selectively activate this subset of neurons. So we can employ this in the same tests that I just mentioned. We can put the animal into an aversive environment. And what we see is that in animals that are controls, where we are not activating those neurons because they've never been labeled, we have normal amounts of freezing or normal amounts of fear. However, when we turn on these neurons, we see a reduction in fear levels, similar to what we see when we put these animals into a context with their cage mate. Now, as a control, we can turn off the light, essentially ending the activation of this subset of neurons. And when we do this, we see that their freezing levels or their fear levels return to that of controls, indicating that it is indeed activation of this subset of socially labeled neurons within the prefrontal cortex that's contributing to this reduction in fear. So this work has a number of potential applications from a clinical standpoint, all of which are still many years away. My lab is currently focused on identifying the molecular attributes of these neurons that make them sensitive to social interactions, as well as the downstream circuitry that contributes to how they modify our fear responses. But one of the things that I want to highlight is that this approach is fundamentally different from that of many other scientists because we're not trying to develop an animal model of a disease. Instead, we're essentially asking the question of what factors, what mechanisms naturally contribute to alleviating fear and stress. And the idea is if we can harness these mechanisms, we may be able to use them to develop augmentation therapies to improve outcomes for cancer and heart attack, as well as develop novel therapeutics for mental illnesses that have been linked to stress and fear, such as PTSD and depression. Thank you.